All right, in this video, we're going to be looking now at the law of independent assortment. Now, with the law of independent assortment, it only is relevant if you're talking about following the inheritance of more than one gene. If you're only looking at one gene, independent assortment does not become relevant. In addition, the two different genes that you are following must be on different chromosomes. So what do I mean by that? Well, here we've got two genes. We've got the Y gene, and they are on these long chromosomes. And of course, we have a diploid organism, and so we have two different Y alleles, or two Y alleles, in this case, two different ones, the little y and the big Y. But then on a completely separate set of chromosomes, we have a completely different gene, the R gene. So the R gene lies on the small chromosome, and of course, because we're diploid, we have homologous chromosomes. The big R on one of them, and the little r on the other. So now, if we wanted, so we're looking at this as it exists on chromosomes in a cell, but if we wanted to look at this with regards to Mendelian notation, what we would write is this: big Y, little y to denote these two guys, and big R, little r to note these two chromosomes here. And what's very important is that the R gene and the Y gene resides on two different chromosomes. And because of that, we say that the Y and the R gene assort independently. Okay, this is really important. So assorting independently is sort of synonymous with the genes lie on different chromosomes. Okay? Now, in order to understand how independent assortment, what it means with regards to the production of sperm or the production of eggs, so the production of gametes, is let's go through here. First, we have to replicate the chromosomes. Boop. All right, so now each one of these chromosomes is duplicated, consists of two sister chromatids. Okay, we still have our homologs. We still have the big red big chromosome and the blue big chromosome carrying the Y gene, and the red small chromosome and the blue small chromosome carrying the R gene. Now, the next thing that we have to take a look at in order to understand exactly what independent assortment means is we need to look at metaphase 1 of meiosis. And at metaphase 1, remember, homologous chromosomes line up as tetrads on the metaphase plate. And so we have this. So the Y homologs pair like this, and the R homologs pair like this. This is where the law of independent assortment kicks in. And what that law says is the way at which different tetrads line up at the metaphase plate during metaphase 1 is independent of all other tetrads, which means it is of equal possibility for these tetrads to line up like this as it is for them to line up like this. Okay, the two possibilities are equally probable, this possibility and that possibility. Now, why is this relevant? Why does this make a difference with regards to the sperm or the eggs that are eventually made from the cell that's going through gametogenesis? Let's take a look here. So here's our one possibility, right? Where we have the blue on the left and the red on the right. The blue harboring the dominant of each and the red harboring the recessive of each. If this is the way the two tetrads line up at the metaphase plate, then during anaphase 1, they'll segregate like this, and then during anaphase 2, and at the end of meiosis 2, you'll have sperm that will look like this. a big Y, big R sperm, and a little y, little r sperm. However, if, and it's of equal possibility, if the tetrads align like this, so that you've got a blue and a red on the left and a red and a blue on the right, then the eventual sperm that you will create is a big Y little r sperm and a little y big R sperm. Okay? And all these possibilities are equal. So it's a 1 to 1 to 1 to 1 ratio. Now, so we can summarize both segregation, the law of segregation, which is a direct result of anaphase 1, and the law of independent assortment, which is a direct result of metaphase 1. And they both have consequences. They both have consequences within how the sperm, what can happen in the sperm. Segregation says any given gamete will only have one allele of a gene. So it will only either have the big Y or the little y, but not both. That's the law of segregation. The law of independent assortment says if you're looking at two genes, then the allele of one gene can be in the same gamete as the allele of any of the other genes, which means we have the big Y, that's one gene, can be in the same gamete as a big R, and the big Y can be in the same gamete as little r. The little y can be in the same gamete as the little r, and the little y can be in the same gamete as the big R. All possibilities are there, and all possibilities occur equally. That is independent assortment. Now, we talked about a monohybrid cross where we're looking at one gene and we're crossing two organisms that are heterozygous in that one gene. A dihybrid cross, now we're looking at two genes, and at both genes, both organisms are heterozygous. So we have big Y, little y, big R, little r. There's the first dihybrid, and we're crossing that by another dihybrid, big Y, big R, little r. And just like we saw with the monohybrid, we're going to get this very distinctive phenotypic ratio. Let's take a look at that. So when we do a Punnett square, we have to include all the different type of sperm. And because of independent assortment, and because of segregation, here are the different types of sperm. Big Y, big R, big Y, little r, little y, big R, little y, little r. And because the female is also the same genotype, then her eggs are also going to be the same. And then we do the same thing. We play this out here. We take, all right, what happens when this sperm fertilizes this egg? Okay, big Y, big Y, big R, big R. What happens when this sperm fertilizes this egg? Big Y, big Y, big R, little r. Okay, and so on and so forth. So, when you do all of this, there are 16 different genotypic possibilities. And when you overlay the phenotypes, what you see is this. You see 9 yellow round. 1, 2, 3, 4, 5, 6, 7, 8, 9. 3 yellow wrinkled. 1, 2, 3. 3 green round. 1, 2, 3. And finally 1 green wrinkled. 1 right here. Now, it's probably better to think about it like this. 9 dominant for one gene, dominant for the other gene. To 3, dominant in one gene, recessive in the other. To 3, recessive in one gene, dominant in the other. To 1, recessive in both genes. Okay? 9 to 3 to 3 to 1. It's something that you'll want to remember. All right, well, what you'll notice is that the more genes you're looking at the same time, the bigger this Punnett square is going to get. And there's going to be a point in time where you simply cannot do Punnett squares anymore because they're far too big and you will never complete a problem on the exam. Which means that instead of completely relying on a Punnett square, you can start to understand the rules of probability. And there are two important rules of probability that you are going to use. The and rule and the or rule. Okay, the and rule. Anytime you say and, you're going to be using multiplication. And I'm going to give it to you in sort of a definition, but then we'll show how you use it in practice. And the and rule, or the multiplication rule, is used to determine the probability of two or more independent events occurring together. Okay, so let's do that. If we have mom, which is big Y, little y, and we've got dad, which is big Y, little y, okay, so we've got two it's a monohybrid cross. What's the probability of obtaining this exact heterozygote genotype? And when I say exact, I mean that a zygote that is little y from mom and big Y from dad. So how do we figure this out? All right. First, what is the probability that this individual is going to get its little y from mom? Well, mom can make two different eggs, a big Y egg and a little y egg. And the chances that she will make a little y egg is 1, because there's 1, out of 2, because there's 2 possibilities. So 1/2. All right. What is the possibility that this person is going to get the big Y from dad? Well, dad can make a big Y sperm and a little y sperm. What are the chances of him donating the big Y sperm? 1/2. So it's a 1/2 chance that this person's going to get the little y from mom. It's a 1/2 chance, and it's a 1/2 chance of getting that big Y from dad. So we're using the and. What is the chances of this happening and this happening? You see how we put the and in there? Therefore, we're going to multiply those two probabilities. The 1/2 and the 1/2. So 1/2 times 1/2 is 1/4. So the probability of the offspring with this exact genotype coming from these two pairs, is 1/4. Now let's say, however, instead we want to find out whether we don't care what type of heterozygous. We don't care. We're just looking at any heterozygote. Whether the little y comes from mom or the little y comes from dad, doesn't matter. Just as long as the embryo that we're looking at is big Y, little y. Now we have something else going on here. So there's two possibilities that fit our criteria. There's one possibility where mom gives rise to the little y and dad gives rise to the big Y. So 1/2 times 1/2. So that probability is 1/4, just like we calculated. But there's another way to do it. Dad can give us the little y and mom can give us the big Y. What's the probability of that? Well, that's 1/2 times 1/2. That's 1/4. Now, this or this fit what we're looking for. They are both heterozygotes, and that's all we're looking for. This or this fit our criteria. We're using or, so we use the addition rule. We add the two probabilities. So we add 1/4, the probability of this, 1/4, the probability of this. Add that up, that comes out to 2/4, which is 1/2. See how we use the or rule there? And we can define that. Used to determine the probability of any one of several mutually exclusive events occurring. If this happens, this can't. If this happens, this can't. That's mutually exclusive. That's what they mean. But either of these two conditions will work for us. This or this. And so we just add up their two probabilities, which equals 1/2. Okay. Now that we have that under our belts, let's take a look at a much more complex genetics problem that we can now solve using our rules of probability And we can solve in a way that takes advantage of the fact that each one of these three genes, the P, the Y, and the R gene, assort independently. They are on independent chromosomes, which means we can treat this cross as three little independent single gene crosses. All right, so we've got these two parents, and we want to know what is the probability of generating an offspring that is dominant, has the dominant phenotype for all three characters, for the flower color, the seed color, and the seed shape. All right, which means what we're looking for is offspring that have a big P and then something. Doesn't matter whether it's another big P or a little p. Big Y and then something. It doesn't matter whether it's a little y or a big Y. A big R and then something. Doesn't matter whether it's a big R or a little r. All right, so what we can do then is we look at this. We look at each one of these independently. If we cross big P, little p with big P, little p, what are the chances? What are the chances that we're going to get any of these three? This, or this, or this? Well, you make yourself, in this particular case, it's a monohybrid. And we already know, when you're doing a monohybrid, you know the 3/4 of a chance that you're going to get a dominant phenotype. Big P, or a big P, little p. So, the chances that the offspring is going to be purple, is going to be dominant for the flower color, is 3/4. But we're not done yet. What we want to know now is, what are the chances that that offspring is also going to be yellow? So let's take a look. When we cross our yellow, we have big Y times little y, or big Y, little y, times little y, little y. All right. So there's two possibilities here. We can have a big Y, little, so this parent here can only donate a little y. Only can make a little y sperm or a little y egg, depending on whether it's the male or female. So the only two possibilities is big Y, little y, or little y, little y. And only one of those is dominant phenotype. So that means 1/2 of all offspring are going to be yellow. We don't know about the purple, we don't know about the round, but 1/2 of all offspring from this mating pair is going to be yellow. And then finally round. Let's take a look. When we look here, this guy can make, half of the time, can make a big R sperm and half the time a little R sperm. Here, this person can only make little r eggs. So again, 1/2 of the embryos generated are going to have the dominant phenotype, round in this case. Okay, so 3/4 of the embryos are going to be purple, 1/2 of them are going to be yellow, 1/2 of them are going to be round, but we want to know what is the probability of this occurring, and this occurring in the same embryo. That's the AND rule. So what we do is we multiply those three probabilities. 3/4 times 1/2 times 1/2 equals 3/16. So 3/16 of the time, these parents are going to give rise to an embryo, to an organ, to an offspring that will both be purple and yellow and have round seeds. That's the way you solve these more complex problems using the laws of understanding the rules of probability. All right, so in the next video, we're going to look at pedigrees, at tracing genetically inherited traits in humans.